Exam 3 is on the skeletal and muscular systems. I think these two are very interrelated. Bones are not going to move without muscles, and we mostly concentrated on skeletal muscles. So, one of the things that you should be able to do is to be able to label a long bone. So, that's one of the first things that we went over was the anatomy of a long bone. So, knowing the difference between the diaphysis, epiphyses, there's also spongy bone, the epiphyseal line, periosteum, endosteum, and then we talked about that compact bone as well. So be able to label a long bone. You can always look on the internet for some different pictures that you can practice with. Another thing that we talked about was the structural unit of compact bone. So, compact bone has a lot of osteons. As you remember, we talked about bone being a composite material. So, it's made out of that hydroxyapatite, and we mostly talked about calcium and phosphate. And we also talked about collagen, which helps to give it that reinforcement and also flexibility. So we looked at the structure of an osteon. I kind of think of an osteon as a layer cake. And if we look at the layers, the cake layer is the lamellae, and then the lacunae is kind of like the frosting layer to me. And that's where those bone cells, the osteocytes, reside. With the communication between the lacunae, you can see those little tiny canals, the canaliculi, and that helps with that communication because the osteocytes, with their outreached almost arms, can sense if that part of the bone is under a lot of stress, and they can send out signals that we need to build up the bone. We talked about that as being Wolff's bone of architecture. So where the bone is getting that stress, we can build it up so that that bone can withstand that kind of stress. They also call this the Haversian canal system because if you look at that central canal, that's where the blood vessels and nerves are going. Along with the osteon and osteocytes that I just talked about, we talked about those different bone cells. Osteoblasts are going to be the bone forming cells. I always think of the bone being born or bring on the bone. Osteoclasts, they have that ruffled border, and that helps to break down the bone, that bone resorption. And basically, hydrochloric acid is kind of pumped into that area. And so that's how the bone is broken down. So the bone is almost dissolved. So osteoclasts, they're the carnivores I always think of chew up the bone. Now, when we talked about the skeletal bone bank being that mineral bone bank, we talked about those osteoblasts when we need to make a deposit into the bone bank. And then we also talked about those osteoclasts when we need to make a withdrawal from that bone bank. The other thing that we talked about was how bone is formed. We talked about the difference between appositional growth. That's when bone increases widthwise, and interstitial growth. That's when bone increases lengthwise. We talked about endochondral ossification. So chondral, that has to do with cartilage, which is the forerunner of bones. And we talked about endochondral ossification being how long bones are formed. Intramembranous ossification, this is how flat bones are formed. This is also what helps with the appositional growth throughout life. So be able to recognize the different characteristics between endochondral ossification and intramembranous ossification. We talked a lot about how to regulate calcium. In the endocrine system, we talked about parathyroid hormone and we talked about calcitonin. In the skeletal system, we talked about how they actually work, and we also brought in the most active form of vitamin D, which is calcitriol. So if you take a look at this schematic, when blood calcium levels are low, this is when the parathyroid glands will release parathyroid hormone. Parathyroid hormone will increase osteoclast activity because now we want to make a withdrawal from the bone bank to put more calcium in the blood. The other thing that parathyroid hormone will do is reduce osteoblast activity, because at this time we're not interested in making a deposit. The other thing that parathyroid hormone will do is that it will talk to the kidneys, tell the kidneys, okay, we want to hold on to that calcium, so don't excrete calcium. We also talked about parathyroid hormone's role in phosphate levels, and that parathyroid hormone talks to the kidneys and says, okay, excrete that phosphate, which helps to lower phosphate levels. So when all of these things happen, this is what's going to increase blood calcium. Now, if we take a look at the opposite, if we have an increase in blood calcium levels, now the thyroid is going to release calcitonin. Calcitonin is going to reduce osteoclast activity, increase osteoblast activity. So take that calcium out of the blood and put it in the bone. So this is when we're making a deposit into the bone bank. So we went through a little summary of calcium and phosphate regulation. So if we're talking about calcium, calcitriol, remember that's the most active form of vitamin D. That's going to raise blood calcium levels. PTH, as we just saw, raises blood calcium levels. And calcitonin is going to lower blood calcium levels. When we look at phosphate levels, calcitriol will raise blood phosphate levels. And the way that calcitriol works is that it has a direct line to the small intestine to help to absorb calcium and phosphate. PTH will lower blood phosphate, and remember, it has that direct line to the kidneys to make that happen. The last thing that we talked about were bone injuries. Now, the good news is that bone is designed to break down and build back up. As we talked about, our skeletal system is under constant remodeling, so our bones are made to heal. Microfractures all happen all the time, and our bones need a way to heal. So we talked about how fractures heal. We also talked about different types of fractures. So the little mnemonic that I use to help me remember bone healing is heal soft into hard bone. So the first thing that happens is we have hematoma formation. So we've got invasion of blood capillaries. Then we have a soft callus form. That's going to be collagen, also cartilage. Remember, cartilage is going to be the forerunner of bone. Then it starts to ossify, and now we get that hard callus formation, and then finally we have that bone remodeling. So I always remember heal soft into hard bone. Then we got into the muscular system, and we mostly talked about skeletal muscle. At the end, we did a little comparison between skeletal, cardiac, and smooth muscle. So the first thing that we talked about is that skeletal muscle is this bundle within a bundle configuration. So around these bundles, we have these rubber bands, as I call them, or connective tissue. So around the large muscle, we have that connective tissue, the epimysium. Then the smaller bundles are fascicles. Around the fascicles, we have the perimysium. And then around the muscle cell, that connective tissue is the endomysium. So starting like superficial and going deep, we have epi, peri, and endo. If we look at the hierarchy, we have the large muscle, then the smaller bundles, which are fascicles, then our muscle fibers, which are cells, also called myocytes. Within those muscle cells, we have those myofibrils. This is also where we talked about those contractile units, the sarcomeres, and then we have those thick and thin filaments that we know as being myosin and actin. So you can see this bundle within a bundle configuration. So this is a microscopic look at our muscle cell. Remember skeletal muscle cells, they're going to be these really long, spindly cells. And then within this muscle cell, we have these myofibrils with these sarcomeres. And remember, sarcomeres are going to span from Z disc to Z disc. And we spent a lot of time talking about contraction and actin and myosin linking up. And when the muscle contracts, it's because all of those sarcomeres, those Z discs, are moving closer together. If we take a look at the anatomy of this muscle cell, we have the sarcolemma, which is going to be the plasma membrane of the muscle cell. We also talked about the sarcoplasm, which is going to be the cytoplasm of the muscle cell. We talked about how the cytoplasm can have glycogen store so that our skeletal muscles get energy. We also talked about that store of oxygen in myoglobin that helps with making ATP. Now, the infoldings of the sarcolemma are going to be the T tubules, and that's where that impulse is going to travel. And then that's going to be associated with that specialized smooth endoplasmic reticulum, which we call the sarcoplasmic reticulum, and its job is to hold calcium. I always say if I had to pick a theme for AMP1, it would be calcium, because we've seen calcium in so many different places. We're especially seeing calcium here in the muscular system. We also talked about it in the skeletal system as well. We also talked about the importance of those sarcomeres maintaining the optimal length, and that has to do with that actin and myosin being in that perfect position so that they can link up and so we can get that good contraction. So we did a little experiment where we use that dynamometer to sort of signify what happens when those sarcomeres are overly contracted or when they're overly stretched. To get the most force, they really need to be at their optimal length. If they're overly contracted, those Z discs are too close together and there's really nowhere for them to go. If they're overly stretched, as you can see with the picture on the right side, then it's going to be difficult for actin and myosin to link up. I gave you the example of how when I injured my Achilles, because of the scar tissue, now my sarcomeres are overly contracted, and that's why I don't have the strength that I once did in my calf muscles. So one thing that you're going to have to do is to put the steps of muscle contraction in order. So I grabbed this picture that kind of summarizes everything. If you can sit with this picture and without looking at the words to be able to go through and explain what's happening, to be able to write it out, then you're going to be in good shape. So first off, we have to have the nervous system. So remember, this has to do with that excitation. So this kind of goes back to what we were talking about in the nervous system, that release of neurotransmitter. So when that impulse travels down the axon, zaps that synaptic knob, we open up calcium channels. So this was the first place that we talked about calcium. Calcium enters that synaptic knob. It causes that cell signaling, which causes those vesicles with neurotransmitters to migrate to the plasma membrane, and then the neurotransmitter is released. In this case, we talked about acetylcholine. Acetylcholine is going to be excitatory on skeletal muscle. Acetylcholine is released, and then it's going to go across the synaptic cleft and then bind to receptors on the muscle cell. Now, where that synaptic knob meets the muscle cell, we call that the neuromuscular junction. So when acetylcholine binds to those receptors, opens up sodium ion channels, now we're generating that action potential. That impulse is going to travel along the sarcolemma. Thanks to those T tubules, now it's going to flow into the cell, zaps that sarcoplasmic reticulum. Now the sarcoplasmic reticulum releases calcium. So we talked about the thin filaments actin. There's two regulatory proteins around actin. Actin has binding sites that myosin will bind to. What's covering those active sites is tropomyosin. What's keeping tropomyosin in place is troponin. Troponin likes calcium, so when troponin grabs that calcium, it moves out of place, which allows tropomyosin to move out of the blocking position. Now actin's binding sites are free, and now myosin can bind to actin's binding sites. Now, if you remember, it takes a lot of ATP to move those myosin heads. So now that actin and myosin are binding, this is when we get that sliding filament theory. It's not that actin and myosin are shortening. It's that as they meet, we're bringing those Z discs closer together and they slide past one another. Now, we don't want the muscle contracting forever, so that means that we have to relax the muscle. So that nervous signal has to stop. We're going to pump calcium back into the sarcoplasmic reticulum. When calcium leaves troponin, now troponin can put tropomyosin back into blocking position. Now those Z discs are going to go back to their resting position and the muscle relaxes. So you're going to have to put these steps in order. So I like this picture because it kind of has everything all at once. If you like to draw, this is a great place to kind of draw things out. If you can explain it, if you can write it out, if you can draw it out, then you're going to be able to put those steps in order. The other thing that we talked about was summation and recruitment, so that our muscles have enough force to do what they're supposed to do. Now, summation increases force by increasing the frequency of the stimulus. This is a little different from recruitment, where recruitment increases force by activating more motor units. So remember, those smaller, weaker units are going to be activated first. Then if we need more force, now we can recruit those larger, stronger units. This is kind of like what I call a friend. When you're moving, and if you've got a couple of friends and you've got a lot of heavy things, we may need to call some more friends in just to be able to put some muscle behind some things and actually be able to move. So summation has to do with the frequency of the stimulus, where recruitment has to do with activating those motor units. Now, it takes a lot of ATP, energy, to contract our muscles. So one of the things that we talked about was where our muscles get energy from. Now, as we outlined in class, the best way for our muscles to get energy is aerobic respiration. That's really the big winner as far as making ATP. So that's the first place that our muscles are going to go. So if we start to work out, in the first few seconds, our muscles are going to use oxygen. However, our cardiopulmonary system hasn't really gotten the memo that we're going to start exercising. So our muscles are going to be getting the oxygen from that oxygen store in myoglobin. Now, that's only going to last for so long, and then we're going to run out of that oxygen. So we still need ATP. So now our muscles are going to go into a more anaerobic means. First place they're going to get it is from the phosphagen system. And I kind of call the phosphagen system nature's Ponzi scheme. This is when we have these phosphate stealing enzymes that are going to be stealing phosphate off of different molecules, pop it on an ADP molecule to make ATP. Like any Ponzi scheme, the jig is up, and that's not going to last for very long. So we talked about myokinase stealing phosphates from ADP molecules, and we also talked about stealing a phosphate from creatine phosphate. And this is why people take those creatine supplements to supply that phosphate to make ATP. Now, because this is going to be short-lived, and we're only about 30 seconds into our workout and our cardiopulmonary system hasn't really caught up, our muscles are now going to be going into anaerobic fermentation, which is what we call the glycogen lactic acid system. This is kind of like where our muscles are stuck in that glycolysis. So since we don't have oxygen as a final electron acceptor, We need another electron acceptor to allow us to make a little bit of ATP. And again, one of those electron carriers, NADH. So it's probably about a minute in our workout that our cardiopulmonary system has caught up with us. And now it's aerobic respiration, which is going to allow us to finish our workout and be able to do like a 3-mile run. So this is the order that our muscles get ATP from. So you want to make sure that you know that order and know a little bit about each process. I am not going to ask you to recount cellular respiration. However, you should be familiar with these. The other thing that we talked about is that some of us are born endurance racers, some of us are born sprinters, and we just happen to have these predominant type of muscles. So sprinters are going to have more fast twitch fibers. So they are going to use that glycogen lactic acid fermentation. So they're not going to have a lot of mitochondria, not a lot of capillaries, but they probably will have a lot of glycogen store so that they can use that glucose. Endurance racers are going to predominantly have slow twitch fibers. So these fibers are going to have a lot of myoglobin for oxygen, a lot of capillaries to bring that oxygen in, and of course, a lot of mitochondria, and probably not a lot of glycogen. So does it mean that just because you have fast twitch fibers that you can't train to run a marathon? It's just that we're all kind of born with these predominant fibers, and they're going to be a little more tailored to certain activities. The last thing that we talked about was a little comparison between skeletal, cardiac, and smooth muscle. Skeletal and cardiac have some things in common where they look striated because of that overlapping actin and myosin. Smooth muscle is not striated, but it doesn't mean that it doesn't have actin and myosin. It's just not arranged in sarcomeres. We also talked about skeletal muscle having multinuclei and many mitochondria. And the one unique thing about cardiac muscle is that it does not require that outside nervous stimulation because those pacemaker cells have that drifting resting membrane potential. However, the autonomic nervous system is sort of like the dials, so that we can dial up our heart rate, dial down our heart rate. We also talked about the difference between single unit smooth muscle and multi-unit smooth muscle. Single unit smooth muscle, this is when we have a sheet of muscles that are all working as one. So we would see that in like the uterus, gastrointestinal system. Multi-unit smooth muscle, that's when they're working more individually. We would see that with like large blood vessels and our airways, so our bronchioles as well. So we did a little comparison and we did that little worksheet with some different statements. So be able to recognize the terminology that we talked about with all three of these muscle types. As I always say, if you can explain it, then you know it. So if you can go through and do a little comparison, maybe even sketch some things out between the muscle types, you'll be in good shape.